Here we have a G protein linked receptor. Now this is called a G protein linked receptor because it works with this protein here, which is our G protein. Now at this point, when the ligand is not attached to the G protein linked receptor, now this ligand right here could be a neurotransmitter, a hormone. It's actually, G proteins are used in a wide variety of signaling situations. It's involved in our smell, our sense of smell, our sense of taste, etc. And so when we look at how this works, the ligand or the signaling molecule is going to attach to this G protein linked receptor. And so this right here is the receptor protein, and when the ligand attaches to it, it's actually going to go through a conformational change in shape. And so what will happen next is here you have your inactive G protein, and we know it's inactive because it has the GDP attached, the GDP nucleotide. However, when this ligand attaches to the G protein linked receptor, the inactive G protein diffuses across the membrane, and it's going to exchange a GDP for a GTP. And now this G protein is active. And I want to point out the G protein is loosely attached to the cytoplasm, to the cell membrane. It's a peripheral protein. It does not cross that lipid bilayer. It's not a transmembrane protein. Anyway, so when that ligand comes off, though, and the signal is done, the receptor protein will actually go back to its original shape. Now, this active G protein, as you saw, can diffuse across and actually have an effect on an enzyme. Here we have, this could be like, for example, adenylyl cyclase, which is an enzyme I use in a different video. And it will activate this enzyme. And these changes in the enzyme will now lead it to take more steps to lead to a cell response. So just changing or activating this enzyme doesn't directly cause a change in response. What we'll see in a different clip is that it will activate and make cyclic AMP as its next steps. However, right here, this G protein is still in its active form right here, and it doesn't stay attached to this enzyme. This is actually how cholera is so deadly, because here the G protein, if it stays attached to the enzyme, in cholera, it will keep the cell response of like diarrhea on, and the person ends up dying of dehydration and loss of electrolytes, all because its G protein stayed in its inactive form. So, lucky for us, once the enzyme has become activated, this G protein right here actually acts as a GTPase, and it's going to hydrolyze its GTP and break it back down into GDP and a phosphate, putting it back into its inactive form. And so now that G protein is back to inactive, and it's ready to have a new ligand come in and repeat the whole process. This also, by going back to its inactive form, this also makes it so that the message is only being sent when there's an actual ligand attaching. All right, and that's the basics of how G protein linked receptors work.